So, hi everyone. My name is Sarah Barga, and I am the research geneticist for the U.S. Forest Service Rocky Mountain Research Station. And today I'm going to be talking to you about a Forb Common Garden study that I've been leading for the Great Basin Native Plant Project. So, currently I would consider myself a plant ecologist, but I actually started working in the Great Basin in about 2008 as a wildlife biologist. But even in the relatively short period of time that I've been working in the Great Basin, it's become really clear that disturbances are increasing in frequency and size. And so here I'm just showing fire perimeters for fires from the last 20 years as an example of a type of disturbance that is adversely affecting native plant communities across the West. And over time, these disturbances add up, causing resource managers and researchers to wonder how to restore native plant communities in these areas of disturbance. So the research that I'm presenting today hopes to get us one step closer to answering that question, as this research relates to the evaluation and selection of appropriate plant materials, which is the second phase in the native plant materials development process. And common gardens are a method for understanding how populations vary in performance across the range of a species. And the Great Basin Native Plant Project is no stranger to this kind of work. They've actually done quite a bit of work with native perennial grasses. And this is a great start because these perennial grasses are important for adding structural diversity to native plant communities. They take up space to prevent invasion. They have low or no seed dormancy, which means that they are predictable in their, once you see them on the environment, they come up predictably. And these plants are also known to be livestock safe forage. But unfortunately, perennial grasses alone don't make a functioning plant community. And so the Great Basin Native Plant Project has sort of turned its efforts more toward looking at forbs in this project that we're calling a Forb Common Garden Array. And the purpose is to take existing knowledge and resources from past experiments and use them to create a standardized protocol to streamline the implementation of forbs into a common garden framework. And this will allow us to determine species-level flexibility to novel conditions, to identify population-level variation in performance in a variety of environments, and to create knowledge and tools for practitioners trying to understand how to select the best seed sources for a particular project. But just to start from the beginning, the foundation of this study was selection of focal species with particular characteristics of interest. So we were looking for species that had a broad geographic range and known importance as resources for wildlife. We wanted species that would be easy to collect and identify by novice botanists, and that were found in large enough populations that seed collection wasn't going to adversely impact those populations. We were also looking for plants that would be easy to produce agronomically. And we ended up focusing on 3 species that include Macaranthera canescens, Canactus douglasii, and Crepus acuminata. And so with the help of several partners, we were able to collect seeds from across the range of these species. And so the map here on the right shows the seed collections from across those ranges. And these populations, those dots represent populations. And these populations were greater than 10 miles apart or greater than 1,000-foot elevation difference. And these represent between 43 and 112 populations from each of these species that ended up in the common gardens that we established last fall. So once these seeds were collected, they were sent to the Forest Service Moscow greenhouse for grow out. And we did this because seed germination can be tricky. And so we wanted to ensure that we would be able to measure plant performance in the field. So they were planted into plugs in the greenhouse. And I want to point out here that even at this early life history stage in the greenhouse, we were able to see population level of variation in morphology in the leaves of the rosette. So here we're looking at Macaranthera on the right and Canactus douglasii on the left. And each of those rows represents a different population. And you can see even with the naked eye, the differences in the leaf morphologies there. So while the grout was occurring, we were busy talking with land managers and researchers to identify locations where we could potentially put gardens on the landscape across the Great Basin. And we found a number of existing exposures as well as other potential sites of interest. And we extracted environmental data from these sites and put it into a principal components analysis that allowed us to select sites that varied along a gradient of environmental conditions and things like annual precipitation and temperature. And so we ended up selecting 8 sites that are depicted in the map in the lower right here across Oregon, Idaho, Utah, and Nevada. And garden installation was truly an adventure and required lots of conversations with people who've done this before. But now that we have a method that we've sort of honed in on, we're working on creating a standardized protocol that we can share with others that are doing similar work. But in general, I just want to give a shout out to chainsaw augers. I didn't even know what they were like a year and a half ago, but they really are the perfect tool for digging like 2,000 holes in a very reasonable amount of time. Another step in this process was deciding what traits we wanted to select to measure in these plants. And we did this by combing the literature for indications of traits that were associated with local adaptation. And we ended up focusing on a variety of fitness and performance related traits, including things like seed weights and inflorescence number, as well as quite a few phenological characteristics and morphological traits associated with performance. So here I'm showing you some preliminary data for average rosette diameter for Macaranthera canescens. And in this figure on the right, I'm really focusing on just 3 seed collections and the performance of those seed collections. And I'm focusing on seed collections that were came from environments that were similar to one of our gardens. So the blue bars represent a seed collection from a site that was climatically similar to Glass Butte. The orange bar represents a site that's environmentally similar to Orvada, and the gray bar represents a site that a seed collection that came from a site that was environmentally similar to Reno. And so in the figure, along the x-axis, you see the performance in those gardens, and on the y-axis, rosette diameter, or average rosette diameter. And you can see that the seed collections that came from an environmentally similar site to a particular garden had a slightly larger rosette diameter. And what you also see is that there's sort of a general size difference in these plants in the separate gardens. So plants in the Glass Butte garden had smaller rosette diameters than plants in the Reno garden. Another question I chose to investigate for this presentation was just whether geographic proximity mattered. And so here I'm looking at the orange bars represent a seed collection that came from a site that was close to our Orvada garden, and the gray bars represent a site that was close to our Reno garden. And again, see that the rosette diameter was larger, even if it was just a slight difference in the garden that was closer to the seed location, the location where the seeds were collected. And so this kind of information, this is just a small picture of like the huge amount of information we're going to be putting into our seed transfer guidance. We also collected leaf tissue along with the seeds in order to perform genetic analysis, and this will allow us to determine whether ploidy will be an issue for these species and will need to be considered in our seed transfer guidance, as well as allowing us to couple lineage data with climate and performance. And so all of this information is going to go into seed transfer guidance to be used by resource managers. And this will include spatially explicit seed zones, like those shown here on the right for blue bunch wheatgrass by St. Clair et al., as well as multiple publications from the Rocky Mountain Research Station, the Ledger Lab at the University of Nevada, Reno, and the Kolmatiski Lab at the Utah State University. And so I just want to give a shout out for Tessa Bartz. She has a poster up that sort of describes the work that she's been doing in association with our common garden work. And this work would not have been a success. It couldn't have happened without our partners. honestly. Like, we received a bulk of our funding from the Bureau of Land Management, as well as funding from the Forest Service and the US Fish and Wildlife Service. We benefited from help with seed collections from the Seeds of Success program and the Chicago Botanic Garden, as well as others. And our seedlings were treated with great care in their rearing process by both the Forest Service Lucky Peak Nursery and the Moscow Greenhouse, and among other resources that we benefited from others. We had 4 gardens on Bureau of Land Management property, 3 on Forest Service land, and 1 on academic property at the University of Nevada, Reno. And we had monitoring help from the Nature Conservancy, Utah State University, and the University of Nevada. And in the future, we're going to be putting a garden in the Ely area, where we will be partnering with the Eastern Nevada Landscape Coalition. Oops. So this year, we're adding a species. We're going to be putting Arid-land fumilis into the gardens, and even as I speak, we have a team in Orovada putting these plants into the ground. And then next fall, we're going to be adding Balsam oryza sagittata and Phacelia hustata. And then as funding allows, we're hoping to expand our taxonomic diversity to include a Lomatium and a penstemon species. And with that, I'd like to thank you for coming and listening to all our talks. And I'd like to thank these organizations for contributing to the success of this project, as well as the help of all these technicians for the installation of the gardens and monitoring of them.